Okay, let's go on the nature of signaling molecules. So we're back to the same 2 dot points. We've talked about the stimulus response model, and now we're going to talk about the difference in how this 3-step reception, transduction, and response, how they work in terms of hydrophilic and hydrophobic signals. Now you'll notice here it says where is the position of the receptor. That is a big part that we're going to be looking at. Where is initiation of transduction? But you don't need to know about these things called G proteins. You don't need to know any names of secondary messengers. And you don't need to know any names of cascade reactions. Okay, so they are not required. So here is your table in your notes, and it is on page 4. Okay, so we're going to look at this table and we're going to look at the nature of the molecule. We're going to look at where the receptors are, and then we're going to briefly outline how they work. Then in addition to that, I'm going to draw a diagram for you. So you may want to pause as we're drawing the diagram. You may want to add this into your notes somewhere. You might want to do it on another piece of paper. So if you need to pause, go and grab a blank piece of paper, you can do that. It's just going to make everything kind of come together at the end, and I didn't put a space for it in the notes. So here is a bit of a summary about what the difference is between a hydrophobic and a hydrophilic response. So this one here is the Now this one is hydrophobic, and this one is hydrophilic. So if you really like this diagram versus the diagram that I'm going to draw, that's fine. You can pause, or you can always come back here and look at this picture here. So first off, we're going to look at hydrophilic because that's the one that's on the left-hand side in your notes. So what are some of the words we can use for hydrophilic molecules? We've already used them. One of them is polar. Okay, so we can refer to the molecule being a polar molecule. If you're a chemist, this means that it's usually got a lot of OHs and a lot of hydrogens kind of sitting off at the side. It's an unbalanced molecule, all that jazz. But as far as we're concerned, this is just a word to use to describe it. The biggest thing is that it loves water. Okay, these are soluble in water. And what this means is they are usually peptide-based or they are proteins. Okay, so protein-based signaling molecules are always hydrophilic. So where does a hydrophilic molecule bind? Think about it. Can it diffuse across the cell membrane? All the way back when we learn about cell membranes. Can it diffuse? No diffusion. So the location of this, that's the wrong picture. We've switched them around. The picture is here. So ignore this bit here for now. So the molecule is going to bind here, okay, on the outside of this receptor protein. It's then going to initiate a bunch of these things called secondary messengers, and they are going to create a cellular response. That could be starting protein synthesis, that could be starting transcription, that could be starting the export of a protein. And so that's where we come back to this one here. So we go signaling molecules. Where are they? They're going to bind on the plasma membrane. They're going to bind on the membrane. They're going to initiate this thing called secondary messengers and eventually bring about a response. And that's it. That's all you have to know about hydrophilic. The main thing they ask you about hydrophilic is where is its receptor and are there secondary messengers? And that is where you go with. Okay, it's always on the outside of the extracellular side of the plasma membrane, or the outside of the plasma membrane, or the surface of the plasma membrane. And then you have the opposite. Okay, so then we start to talk about hydrophobic molecules. So now ignore this picture because it doesn't apply to this one, and I'm going to bring them back together again in a second, and I'll go through them on one slide. But hydrophobic ones are non-polar. That means they're insoluble in water. And to be insoluble in water, what does that mean you are? You are probably a lipid, you are probably a fat or a steroid-based hormone. Now, what happens with these guys, so this is this side here on the left, is they can directly diffuse through the cell membrane. if they can directly diffuse through the cell membrane, there's no need for any of these secondary messengers. There's no receptor protein on the outside of the cell here. Instead, the receptor protein is somewhere in the cytosol, or possibly it's even in the nucleus. So that molecule can diffuse into the cell, pass all the way through, and bind to, so here's your signaling molecule, it binds to a receptor inside the cell somewhere. So that's an intracellular receptor. Now that molecule, that signaling molecule and the receptor are now a new molecule. And what that does is it moves along, and usually what it does is it acts as a transcription factor. So if you need to pause and go back and think about what is a transcription factor, you might want to go back to the notes from Area Study 1, the third part when we talk about gene regulation. Transcription factors can either switch on or switch off genes. So what we've just done is we've made a transcription factor. Now that transcription factor is going to activate or stop protein synthesis. So that's its cellular response. So your reception is this first bit here where the signaling molecule binds to the receptor inside the cell. The second bit is it moving within the cell. The transduction is it moving within the cell. And the response is starting or stopping gene transcription. And that's kind of a comparison of the two. Now, I'm sorry the pictures were a bit mucked up. I'll fix them on your PowerPoint before I post the PowerPoint. Now, the last bit is signal amplification. So this is a really small part of the course, but often it can be an exam question that causes anxiety. So this is the fact that you get one signaling molecule, and that one signaling molecule triggers many secondary messengers. Can you see here that there's only one signaling molecule? I'm just going to change my color so that you can see it a bit easier. There's one signaling molecule right up the top here. So it binds to its receptor over here at number 1, and it initiates this series of secondary messengers. But because it's bound to one spot and it doesn't just leave straight away, heaps of secondary messengers just keep coming up and getting activated. Then each one of those activates many. Can you see how that one signal has created all of this response? So that's all we need to say. If the question asks you, how can one molecule bring about a rapid cellular response? This is how. Is that one signaling molecule can activate many secondary messengers. And that's all you need to say. So that is the end of signal transduction.